we will be talking about how the read counts was generated from the beginning of the Fastq files, how the filtering and the trimming was done before it arrives at the final count matrix for any follow-up analysis. So first, let me give you an introduction, quick overview or review of the 3' single-cell RNA-seq technologies that was introduced by Adam in the previous seminar. So here in single-ROM, the single-cell RNA-seq was performed on the microchip and the microvials. Well, in each of the vial, a cell is captured, and then in each of the cell, one bead occupies the space with different sequences or molecule components attached to the bead. So in each bead, it is connected to the molecule sequences where it is composed of the PCR handle, the cell barcode, the UMI code, as well as the poly T tails, so that in each bead, the cell, a unique cell barcode from all these molecules will be attached to the bead, where all of the molecules with different UMI counts can be attached to the bead and return with the different arrays that is mapped to the poly T tails here. So this is a schema that explains the read constructions from the read one on the left side, and which connects to the poly T tail by matching the poly A mRNA tails, and then it also includes the information of the cell barcode and the unique UMI for each molecule. So on the reverse side, with the inserted sequences, we will see the read2 sequence also can be transcribed here and then sequenced to give about 150 base pairs of the sequence lengths for the read2. And for read1, various lengths can be observed from different platforms or different device companies. So here we recommend a minimum 72 base pairs for read1. Sometimes the length of read1 can go as high as 150 base pairs to capture more sequences. So here it is a schematic view of the components or the structures of the read1 versus read2 after we obtained the information from the sequencing companies. Okay, so the next part, we are diving into the single-cell data pre-processing workflow. So the pre-processing workflow is composed of these few steps. So the first step is we obtained the Fastq files. Normally, there are 2 Fastq files for each samples. And then from these Fastq files, the demultiplexing can be done to group all of the read sequences from the same samples into individual Fastq files. The next step will be the trimming, where the low qualities component of the rays or the poly T part of the rays will be trimmed off. And with the cleaner rays, the alignment will be performed, followed by from the alignment, we will know which gene these UMI molecules map to. And from there, we can calculate the feature counts where for specific for single cell, where the genes, a particular gene, as well as a particular cell can be determined, so that for each individual cells, we know the gene expressions for at the individual cell level. And finally, the count matrix are generated from this whole process. So here we are going to begin from the the explanation of the Fastq files. So here in this example, we are going to going to drill down and focus on this one particular Fastq read, the read sequence. So this read sequence will follow us from the very beginning where it first appeared in the Fastq file until the very end. It goes into the BAM file, and eventually it will be counted toward the count matrix. And I saw a question from Habiba just now. So why does the minimum 72 base pair is needed? So it's actually a good question. So because here, if we don't use the poly t-tails from the read one, actually if only the sequence 60 and 12, it is long enough to form some read sequences that can support the uniqueness of the cell barcode plus the UMIs. So in Stethoscope, where the single runs processing pipeline, there is a specific parameter can be tuned so that we can enable the mapping without the poly T sequences. Okay, so let's go forward. So this sequence here is a typical sequence in the Fastq files. In the Fastq file, we will see many sequences where this is one of them. So for each read, this represents a read in the Fastq file. In each read, it always starts from, there are always 4 lines. And the first line is the header line. It starts with this @ sign, followed by the unique description ID, where there are 2 parts of the unique description ID. The first part is the instrument. Here is A00159, followed by the run number. The run ID is 607. The flow cell ID from the instrument company anomalies from Illumina is these characters. And then followed by the lin number, the tiles, as well as the tile coordinates, so the X coordinates and the Y coordinates. So the first part, description ID, actually already uniquely identified this read, that this read is unique to itself, and then it is followed by the sequencing ID. So here bolded as 1. So 1 means this is read 1. If it is read 2, you will see a 2 here. N means this read passed the initial sequencing quality control. 0 means this is not a control read. So in most of the next-seq experiments, like we only observe zeros here at this position of the sequences because no control read was used. So it means this is a real true read from the biological experiment. And the last part is the sample index numbers, character strings. So from here, we have the header that is unique. And the second line will show the ATGC sequences. Basically, it's about 100 to 200 base pairs long. Like mentioned earlier, it can also be 72 base pairs if we only capture the first part where it includes the cell barcode as well as the UMIs. So the third row will show the plus sign, or alternatively, some Fastq files also have the plus sign together followed by the description IDs. And the last row of this read is a string of read quality scores where it indicates the it represents the percentage of having a wrong nucleotide at a specific position. So normally, so if the score is more than 20, means error rate is 1%. If the score is 30, means the error rate is 0.1. So a lot of times we will see the Q30 quality matrix is reported for the sequences to indicate the quality of the Fastq rates. So the Pareto score is for each nucleotide, there is one score alphabet that represents the score here. So the next here, it is a table of the corresponding alphabets with the probability of error here. So yeah, I saw there's a comment. Thank you for the comment, Mahab. Yeah, so here I think I meant the ASK2 score, for example, previously from the previous slide, we see F. So F here means the error rate of 0.0002, which means it's a relatively small error rate. And from the sequences we saw just now with comma and with the colons, so the error rate are like 0.003, and with comma, the error rate is higher, is 0.007. So yeah, so okay, so it can be indicating the probability of error, or it can be indicating the qualities. So I guess it depends on whether we are talking about the P error or talking about the Q. Yeah, it's a good point. Thank you. So from here we observe, okay, this sequence, most of the alphabetic quality, the Pareto score here is F, which indicates this rate is of relatively good quality. So let's see what it will become in the next steps. Okay, so here, yeah, let's go back to see like what are exactly read 1 and read 2 and how are they reflected with regard to the nucleotides in our Fastq files. So the read 1 will have the 5' adapter. It will contain the cell barcode, the UMI, and the poly-T. How about read 2? Read 2 actually contains mostly the insert sequence information. So how is it represented in the nucleotides here? So this is our read 1 in the R1 Fastq file. And this is our read 2. So we can see it is actually forms a pair. So we searched for this read in both of the Fastq files in R1 as well as in R2. So we can see here the description ID of this Fastq read is the same for both of these two reads, indicating they are the same pair. And then they are comes from the same samples with the same sample index, and they both pass the quality filter from the sequencing machines. And then we have R1 and R2. How about the where are our UMIs and the cell barcode? So in Stethoscope or single runs platform specifically, we can see that the UMI follows immediately on the left side from the poly-T tails. So the blue color here represents the UMIs, and the cell barcode are split to 3 different components, with each of the components having 8 nucleotides, and then all of them together will have a 24 base pair of cell barcode. And then here, the black color in between of the cell barcode, they are the linker sequences. So the linker sequences help the bead, remember there is a bead on the left side to continue synthesize this sequence. And then later, we can make use of the linker sequences to remove the read 1 with lower qualities. Okay, so yeah. Okay, so yeah, we are able now to map the cell barcode with the UMI as well as the poly-T tails. So here, from the read description, we know read 1 and the read 2, they are from the same pair, which means we know they are together. But the thing is, read 1 has this UMI and a cell ID, but read 2 don't have it. So for now, for this one, specifically speaking, this fragment is long. So the read 1 and the read 2, they don't overlap. In some locations, we do observe the read 1 and the read 2, they can overlap, but for this one, it doesn't. So, and then the sequences that we are really interested to count are the sequences from read to, actually, because the read to represents the insert sequences. And then this is the read that we actually map it to the genes or the genomes later. So the next step is a demultiplexing step. So in this demultiplexing step, the Fastq files are cleaned up in a way. Each read from read 2 is renamed according to the cell barcode and the UMI determined by read 1. So here, this is a real Fastq files. Well, you can, if anyone is interested in test run, this can be downloaded from the Cellscope GitHub repository where you can see the Cellscope test data. So after the step of demultiplexing, now we obtain one Fastq file. So compared to previously, we have two Fastq files. Now we have one Fastq files only, and then it only contains read 2. So in this read 2, each of the names of this read is updated. So that it only includes information about with the cell barcode as well as the UMIs. And here for this particular read, you can see it appeared 2 times. So with the same UMI, with the same barcode, it's only one molecule, but it appeared 2 times. So in this table summary, you can see, okay, one barcode, one UMI. Okay, later it will be aligned to a gene, but now we don't know yet. And then the UMI count is 2. So it may later in the next few steps, in the feature counts here, we will solve this problem by deduplicating these UMI counts from duplications. And in this step, there are a few filters applied to read 1 as well. So for those reads without the linker sequences and those without the correct barcode, so for the read 1 without proper poly-T tails or the low quality reads, they will all be removed so that the corresponding read 2 will not appear in these Fastq files. Then the Fastq files only contain a clean version of the read 2 that is truly representing the true reads. It is not come from the low quality cells or the cells that doesn't bind to a valid barcode. So in actually the both the Cellscope as well as Cell Ranger. So for those common single cell processing, pre-processing pipelines, there are some steps to do the batch code, the cell barcode correction, as well as the UMI corrections. So normally the UMI added distance of 1 or the cell barcode added distance of 1 are allowed to allow like a one nucleotide mismatch between the UMIs as well as cell barcode to enable the tagging of the read to the correct UMI as well as the cell barcode. Okay, so now we obtained the read from the true insert sequence reads, and then the next step will be the trimming. So the trimming will remove the adapters, any adapters that are potentially still remains at this step, and it will trim off the poly-T tails. Also, a lot of times we will see following the poly-T tails or at the sometimes towards the end of the read, we can see the quality becomes lower. So all of these bases will be trimmed from read 2, so that the reads can be smaller and cleaner, and only the insert read that is truly representative of the genomic sequences remains after the trimming processes. So there are some trimming tools. The one that are currently most used in many of the pipeline algorithms are the cut adapt. So it's used in both Cellscope as well as Cell Ranger. Trimomatic is another popular tool. So trim galore is also use the cut adapt at the back end to do the sequence trimming. So here again, we see the two read here, 8591 and the 9093. So these reads are still with us. So we can see that this read was not very much trimmed, because from the previous step, we see the read quality for these two reads are actually quite okay. So they didn't go through they went through proper trimming, but they were not over trimmed. And then bottom left also shows that okay, so in these trimming processes, we do sometimes observe the sequences with adapter sequences, like for example, for this particular test, 26.96% of the reads, they were having the adapters, and then these adapter sequences, they were trimmed off. So, and then the low quality base pairs, 8% of the low quality base pairs of the overall bases, they were low quality, so they were also trimmed off. And from here, after we obtain the cleaned, trimmed reads, now we can align these reads to the reference genome. So the first step to for the alignment is normally to create this reference genome, which is represented by the genome indexes. So it was made from the FASTA nucleotide sequences, and then here this example actually comes from in the ensemble primary assembly FASTA file. So this is one of the recommended assemblies to go with. And then the GTF also comes from the ensemble. So normally when we do these make references, we make sure the FASTA files as well as the GTF files, they are of the same version, or they come from the same resources, so that the positions, locations of the genes can be mapped. So the FASTA file basically includes the sequences, the complete sequence from the different chromosomes. And the GTF files are rows of genes and its locations on the chromosomes, as well as the coordinates. So if we want to do any inclusion or exclusion of a certain genes, we can do it at the level of the GTF. For example, if we don't want the pseudogenes to be aligned as part of the reference genome, these pseudogenes can be removed by processing the GTF files. So eventually only the genes that are exist in the GTF files that mapped to the primary, the FASTA sources, these genes will be looked at and counted towards later. There are several alignment tools, as Yasmin first introduced. So here we briefly talk about 5 of them. There are several review papers actually compare different tool performances. So they are good review papers. So if anyone is interested, you can go and take a look. Basically, I think the main conclusion is the STAR. So STAR is used both in Telescope as well as Cell Ranger. So this is a reliable alignment tool. It is relatively fast compared to the other tools. It produces more genes, and then for each of the detected genes, it has the higher expression values that is accounted to its genes. So STAR Solo is a more recent variant of STAR. It is faster than STAR, and it has some additional benefits. For example, it can identify the splicing junctions very well using this tool. And but the limitation of STAR, one of the limitations of STAR is it requires relatively high memory resources. So it will require 32 GB of RAM to process in a relatively good time frame. So some of the other tools that are also used sometimes in the field are HiSec Set2 It is based on the graph index calculations. This tool requires relatively lower memory resources, but it can be relatively slow. And the Bowtie or Bowtie tool is also index-based aligner. It is fast and low memory resources. It was also used quite often last time for the Box-seq sequencing alignment. But then Bowtie 2 is not aware of the splice, so it's not as optimal for the RNAs. So it's more for the genomic sequences where not so focused on the transcriptomes. Well, recently there are some new alignment tools like Callisto as well as 11Fry, like they are alignment-free tools, and they use pseudo alignment. Basically, is a K-mer-based alignment that generates the clusters of the K-mer sequences and also map back to the reference genomes. So both of these tools, they are fast. And then for Callisto itself, it is specifically also very efficient. It's one of the fastest alignment tools. But for Callisto, there is sometimes the review paper reported that it can introduce artifacts, those genes that are not supposed to be seen in some of the sequencing runs. They still call them positive. And for the LEV-Fry, it has for some of the samples, it cannot detect the lower expressed genes as effective as the other tools. So overall, we would still recommend to use STAR or STAR solo for the mean sequencing alignment tool if you have sufficient memory resources. Otherwise, if there is a limitation of the RAM or computational resources, you are not very particular about the accuracies of the lowly expressed genes or small artifacts, maybe you can try the 11Fry, Callisto, or HiSet tool. So here is just to mention a little bit about the STAR, because it was used is quite a popular tool with regard to the gene sequencing alignment for single cell as of now. So it is actually based on the maximal mappable prefix that will be searched against the created genome index, which is represented by the suffix arrays. And when the first time during the search, the algorithm will look from the leftmost side, the first nucleotide of this RAID, and then it will find the longest exact match from this RAID to exon in the genome until the time where it cannot map anymore. And then it will stop here and map again from the leftmost locations of this RAID, and then to look for taking the MMP1 as the anchor to look for the remaining sequences in the nearby exons, and to try to map again. So sometimes it can stop multiple times, and then look for, so it's possible to allow some mismatches where this process start and stop and start and stop several times. So eventually, after we have these mappings from different iterations, it has a clustering process to group all of the identified seeds, aligned seeds, with some stitching, and finally there is a score from the local alignments to take account of the mismatches, insertion, deletions, and splice junctions, so that we have a quantitative assessment of the alignment qualities and the ranks, and then the rates can be assigned to each of the exons or genes accordingly. And it depends on different alignment algorithms. So different algorithms actually handle the rates mapped to the multiple genes differently. both in Cellscope as well as Cell Ranger, these two major pipelines. The reads that map to multiple genes, that is usually called the ambiguous gene read mappings, they were discarded and not used in the later feature counts. But in some of the other algorithms like Alivin, actually one read, if it mapped to multiple different genes, this one read will be split into different parts and account for the counts from the multiple reads. Okay, so after the alignment, we now obtained the BAM files. So it's another file that structure that captures the result from the sequencing. So again, we see these two sequences. So they still follows us from the Fastq files to trimming to the alignment. So here in the BAM file, it has the read name. Again, take the cell barcode as well as the UMI, and then it shows the chromosome where this read aligns to, the position of the chromosome, as well as the read sequences and the PRIT scores. So here the PRIT scores will indicate the quality of the mapping. And with some of the other texts towards the end shows additional information with the mappings, which we will see from the next slide. Yes, so from the alignment, here from the alignment and then from the genomic locations that this read managed to map, we can now go back to the genomic locations of the genes. So here, now, after the alignment, we can start to count the features. So the reads are now assigned to features by using their mapping position to the reference genome, and then the uniquely mapped reads to the genomic features are extracted in a new BAM files. And then at the last, the sum of the unique UMI counts on each cell barcode are calculated for each of the gene at this step. So going back to the Fastq file, BAM files, going back to the BAM files. So if we, like, for example, use this command line to search for our read name, we will be able to find these reads in the BAM files. And then additionally to the sequences now, we have more information, like the CB here is the cell barcode, the UB here is the UMI sequence, the GN is the gene name, and then we also have the GX, the ensemble gene ID. And also additionally, with regard to the sequence mapping, we have the here NH, the number of reported alignments that contains the this sequence in the current record, and then the ICE alignment, the alignment scores, as well as the the edit the NM is the edit distance to the genome. So here is 0 means it is like a full 100% match with the genomic sequences. And then yeah, the XN is the number of ambiguous bases in the reference. So which means in the reference we may have a N or some other nucleotides as the sequence other than the ATGC itself. Okay, so now we are able to, we have from the sequences, from the raw sequences, we have the information of the genes. So the next step is to count them. This is a representation of the final gene matrix that we normally obtain from after the single cell preprocessing workflow. Here, the UMIs, they are counted for each feature, which is each gene, and each barcode, which is each cell. So there are two formats. One is the sparse format. So another is the full matrix. So just now we saw that this rate using from this gene, this FIGN gene, using with this barcode, there are two rates. But then eventually when it goes to the UMI counts, only the unique number of the UMI is counted. So that we only report one as expressed in both the sparse matrix as well as the full matrix. Actually, these two matrices, they represent exactly the same thing. The only difference is the format, reporting format of these two tables. So in the sparse matrix, the counts represents the okay, so the three different columns are shown here, with the first one column being cells, another column being gene, and the last column being the count. Here is the number of unique UMI counts for each of the cell barcode plus the gene combinations. Yeah, so the benefit of sparse matrix is it does not need to have all of the pairs in the full matrix to appear. Like for example, you can see the one here, but then like the cell 1 and the gene 2 will not appear in the sparse matrix because it is 0. And it is specifically useful because normally the single cell experiments are quite sparse. And you will have typically 30,000 plus genes, and then with a lot of them being 0 counts for the genes. Having a sparse matrix is helpful to reduce the size or the volume of the read counts, and then easier for processing, and good for the computational calculations later if there's any follow-up analysis. So the next part, after we obtain this count matrix, we'll, like some of the cells, for example, this cell 5 here, this is an example, but it just mimics the condition where sometimes these all of the genes are 0, or the genes are actually very, very low here, then it doesn't support cell 5, although with a barcode being a true cell. So like some of the cells in the count matrix, they are not cells because of their low total UMI counts. So in the count matrix process, we will do a cell calling. So there are multiple cell calling algorithms in different pipelines. So the recent one are using the empty drop method. So where the initial cells are calculated, and then empty drop was also calculated, and then a stratified barcode of empty versus cell-containing barcodes are calculated on the remaining cell barcodes. So this has been implemented in both Cellscope 1.10 onwards, as well as Cell Ranger 3.0 onwards. Okay, so for the cell calling, just a little bit of details. So the algorithm will determine the initial cells from the barcode with 10% of the top 99 percentile of the total UMIs. And then it will construct a background expression profile from barcodes with a very small UMIs. So with a definition of very small UMIs, you will be able to estimate ambient RNA profiles by calculating the model fit from the multinomial distributions using the simple good Turing algorithms, where from this you are able to infer and determine whether cell barcode are they sufficiently distinct from the ambient profiles, or are they still closer to the true cells that was determined from the first steps. And yeah, from here you can assign or calculate the cell barcode to being a true cell versus possibly they come from empty empty well. So yeah, it has been used in the newer version of both Cellscope as well as Cell Ranger, and I think in both Cellscope as well as Cell Ranger, the true cells as well as the ambient empty wells, they were reported. So here, this is the final part. So it covered from just now, we covered from the Fastq files until the count matrix from the very beginning to the part where we obtained the count matrix. Here is just to give you some additional information with regard to if anyone wants to try the code by hand. So these are the steps of the code, and the output you will retrieve from running these different steps of the code. And the examples are extracted from the Cellscope Quick Start website. So actually the Cellscope itself as a pipeline tool, it is a one-step tool will automatically generate all of these steps for your analysis. So you do not need to generate all of these steps by hand. You only need to run the Cellscope at the top level, and all of these commands will be generated for you. But here we just still show the command just for the purpose of explanation, like what happens at each of the steps, so that you have a better idea of what has been done and where to look for results, or if later you want to do something different, like for example, you want to replace the alignment tool with another tool, is it possible in Cellscope, and which step you need to change? So here, the first step, the deep multiplexing to generate read two only Fastq files, you can run the Cellscope RNA sample and the Cellscope RNA barcode. It will return you a Fastq file with only the read two, the R2. And here it also, this is part of the Cellscope report. It also reports the quality matrix or the results from demultiplexing. Like for example, it tells you how many raw reads there are and the percent of the valid reads and the Q30 of the barcode, as well as the UMIs for your QC report or any processing references. And the next step is to generate the trimmed Fastq files. Here you can use, as you see, the program use cut adapt. I believe the Cell Ranger actually use a similar trimming software here, and it will now give you a clean read to only Fastq files where you can identify and determine different parameters like a minimum 20 base pairs, so that if the rate is below 20 base pairs, it will not be discarded after this step. And then here the next secret trim is it will remove the trim off the poly G tails that is comes out from the two color processing in some of the next-seq or the nova-seq sequencing steps. Yeah. So here it also tell you, okay, so how many rays has been trimmed with adapters, and then how many rays are not used because it's too short, and how many final rays you will have. So everything will be reported at this step. And then the next step again is the alignment. So here we can see it used the star alignment, and it will report the initial BAM files without the mapping to the genes. So it will only tell us the genomic location of the mapping of the each of the rate, and it tells us how many of these rates are uniquely mapped, and then how many of them are mapped to the exonic region or intronic region or intergenic regions. The next step is it will count the features. Now in this BAM file, we begin to see the gene names according to it. And then, yeah, again, it has this feature count quality matrix to tell us how many rates are, or base pairs, how many base pairs are assigned to a specific gene. And then it determines the number of cells in the next steps. So it depends on different versions. So in the older version of Cellscope as well as Cell Ranger, it used the knee plot to determine the cutoff of the cells. So the knee plot actually take the midpoint of the first fall of the barcode rank plot. So normally this UMI count versus the sorted barcode counts, it has two sudden drops as represented by knees. So the knees will be determined here. The cutoff will be determined at the middle point of the first knee. So in the later algorithms, the algorithm changed to make use of the ambient RNA arrays, but still, I think the barcode rank plot are still reported to give us a look or the inspection of how the cell calling algorithm works. Yeah, and the last part is the analysis. So we will not cover analysis in this talk. In the next few talks, we will be covering more about the analysis. Yeah, so we look forward to the next talks as well with regard to the analysis. But from now, we have successfully processed the Fastq files and obtained the count matrix where we have get both the cells as well as the genes. Yeah, so in summary, there are several ready-to-use pre-processing pipelines, including Cell Ranger as well as Cellscope. So you can choose the right tool based on the available resources, like your computational resources or the speed you require, as well as how large or small a data set is. And then the cell calling sometimes is highly influenced by the presence of background. So it is actually important to have a clean library to go forward. Yeah, so let's conclude today's webinar with regard to, please feel free to contact info@singleroundbio.com. This presentation has been co-prepared by Adam and me, and special thanks for Jonathan, Huiqi, Wangxi, and Yingting for the help with regard to these presentations and general processings. Thank you.
Thank you, Stacy, for the very interesting talk. So we can start our Q&A section. We have plenty of questions here. So yeah, let's start one by one.
Okay.
Relating to Fastq files, does the range of quality scores depend on the sequencing platform used to produce the Fastq file?
So, okay, let's go back to the Fastq file page. Right. I think it's a very interesting question. I cannot say for sure, but I think it's a good question to check whether the quality scores, for example, is determined by the sequencing platform or sequencing companies. For example, if we re-sequence the same rate again using a different platform, will we have a different quality scores that is associated with this rate? We haven't done a thorough review with regard to the Fastq sequences, but I suspect it may depend on the for example the sample qualities, like how strong the fragment you have with regard to when you for example, we'll select and cut these insert sequences, like how strong or how clear each of the fragments are, or are the fragments here like flying around that is causing too much to introduce some noise to the sequencings. So it may has some influences on the Fastq file qualities. So we have not done a thorough comparisons or a survey for the rates with different sequencing platforms. I cannot say for sure, but I suspect the Fastq sequences may depend on multiple factors that is the sequencing platform or sequencing company may be part of it. Like, for example, if regardless of the single cell or short read or long read, different sequencing technologies may introduce different error rate, for example. When this happens, we may expect different quality scores returned from different Fastq files. Yeah, I hope that answers.
Next question is about referential compression. So one of the biggest challenges in referential compression algorithms is to efficiently find long matches between the reference and the read sequence to be compressed. So once matches and mismatches are determined, another change is to find a space-efficient encoding scheme. Any advice for that?
Sorry, I didn't get the question. So do you mean different splicing variants that is captured by the different alignment algorithms?
Maybe we can, if the person writes us, we can answer this question for sure. Another one is that, does the remaining sequence after poly T of read 1 be applied to read mapping step 2? So read mapping step 2, or just skip them?
So the read one will be not be used for the sequence matchings. So because here we can see sometimes the fragment can be long. So the read 1 and the read 2, they don't meet each other. Then here it may be slightly difficult to use both read 1 and the read 2 to map to the genome. Sometimes if the insert sequence is short, they do overlap with each other. Then there's another scenario like, okay, it may be used to improve the quality of the cell calling, but at this moment, all of the sequences after the T, the poly T here, they are not used. And the part of it, I suspect, is because sometimes for read 1, after the poly T, the quality scores, we will see a drop in the quality scores. So many of these, the later part of the sequences will be trimmed off anyway, so we may not be able to use them. So in short, we only use for read 1, the main purpose of read 1 is to give us this identification of the barcode plus the UMI combinations. Yeah.
Yeah, so the process of demultiplexing and Fastq file generation can be very slow. Is there a faster and more efficient platform or approach to use?
Okay, that's a good question. So far, I think it's yeah. So far, the process is slow. Part of it being the process, like it will need to look at the different algorithms or different parameter settings to change the map by the edit distances for both the UMIs as well as the barcode. So here I don't use the any different algorithms to search for the UMIs as well as barcode, but I believe this may be able to overcome by use a more like speedy algorithms to maybe, for example, to have some greedy search methods to search for the UMIs as well as barcode for the identification of the match with the reads. Yeah.
So is the barcode sequence divided into 3 sections? If so, why?
So here in this platform, it is divided through to 3 sections. So the main reason is first to enable the bead to continue synthesize by having a linker sequences here. So have a stronger affinity with regard to 3 different components of the barcode. And the second part is by having these linker sequences, it will enable the as part of the quality control as well. So if we don't observe this linker sequence, means this read one is not properly generated, and then the read one will not be used for the later feature counts.
And is the pre-processing workflow is the same for single cell sequencing and bulk sequencing?
They are different. So this specific workflow is for single cell sequencings. As we see, I think the additional steps are the demultiplexing, where it is assigned to a specific cell barcode on the left side. So for bulk-seq, there's no specific cell barcode that is needed. And also, okay, I think the trimming may be the same. So the trimming may be the same if you know like which adapter to be trimmed. The alignment part will also be similar as the, like, for example, many of these tools like the STAR as well as Bowtie 2, for example, they were actually developed from the bulk-seq. But then the difference is for single cell, eventually it will need to count against one cell barcode. I think this is the unique part for single cell, where you will need to have this cell barcode in order to calculate how many genes are expressed here. So there will be a difference in the single cell versus bulk-seq pipeline processings. Yeah.
Yes.
And
so, yeah, sorry, go ahead.
Yeah, I think this may be one of the time consuming part for single cell. I suspect if for bulk-seq, without this step, it may be slightly faster. Yeah, but you lose the ability to assign the UMIs for each of the cell at the cell level. So many of the downstream analysis, for example, will depend on having the ability to have these different cells and the ability to group them into different subgroups to conduct some further analysis and then to derive some of the insights to your sample and the data.
Thank you again and have a great day.
Thank you, everyone. Thank you, Yasmin. Bye.
Thank you. Bye.